Hi, my name is Alexandra Dark, and this is our group experiment project for biology. Our experiment is called Dirty Hands Experiment. Our group is called the Genetic Diseases. Um, my name is Alexandra Dark, and my co-authors are listed here, Mariah Rose Samir, Benjamin Leba, Bianca Acosta, Aria Williams, and Gage Serberg. We are all students here at the University of New Mexico. The background to this experiment started really with our desire to look at what is the best way to keep our hands clean given the COVID-19 pandemic. So we looked at the CDC guidelines on hand washing, and we found that by washing hands with soap and water reduces respiratory illness like colds and upper respiratory illness in the general population about 16 to 21% from the CDC guidelines. The use of hand sanitizer can also reduce the number of germs on your hands if there is no soap and water present. You want to make sure that you use the hand sanitizer that is at least 60% alcohol. From the Module 3 articles, we were able to design our experiment. What we found from those articles was there was really no difference in the use of antibacterial soap versus regular soap. And soap is superior to the use of hand sanitizers. And cell phone is actually 10 times more dirtier than a toilet seat. So it was important to us to keep our hands clean in order to reduce the spread of COVID-19 virus during this pandemic. So our question for our experiment was, what is the best method for disinfecting hands?
Our null hypothesis for the experiment is that hand washing is not the best method for bacteria removal, and hand sanitizer is. The alternative hypothesis for our experiment is that soap and water is the best method for removing bacteria from our hands. And our prediction for the experiment is that the hand soap treated bread will be fresh and without any mold. And we thought that the one treated with dirty hands would have mold and bacteria on it. The materials that we needed were Ziploc bags to store the bread in, sterile gloves so that we didn't put any bacteria from our hands onto the bread, and sterilely white bread, soft hand soap with aloe vera, Ultra Defense antibacterial hand soap with hand sanitizer with alcohol, Dawn dish soap, black Sharpie, and commonly touched surfaces such as phones, laptops, light switches, or door handles, or things that carry bacteria on them.
So the methods that we used for this was, our step one was we prepared each bag by labeling the categories with the dirty hands, hand sanitizer, the no touch, and then the 3 types of soap, as well as the date for each. And then next we put on the sterile glove and refrained from touching any of the surfaces and placed a piece of bread with the gloved hand in the Ziploc, and that would be the bread, which was the control for the no touch. And then the next step was I touched commonly touched surfaces around the household, such as phone, laptop, light switch, and door handles. And then once I did that, I touched all over the bread so that it would be treated. And this would be the dirty hands treated bread. And after that, we placed it in the pre-opened bag and sealed immediately. Next was the hand sanitizer, making sure to coat the entire hand evenly and thoroughly. And after doing so, put the bread in the bag similar to the dirty hands one, and then sealed it. After that, I retouched all the same surfaces used for the dirty hands treated bread, and then used the hand soap to clean the hands and make sure to properly wash the hands for the typical time that was recommended for the CDC, which was 30 seconds. And after washing the hands, I grabbed a piece of bread very similar to the hand sanitizer. And then after that, step 5 was repeated for the antibacterial soap as well as the dish soap, including touching the same surfaces and washing the hands for 30 seconds. And once all the bread was prepped, a chart was made to see the growth on the bread. And then I placed the bags in a safe area away from any animals or things that can contaminate or affect the experiment, such as temperature or light.
So these are our results, and as you can see with the table, we do have the 6 different pieces of bread, varying from no touch to dirty hands, to the use of hand soap, the use of antibacterial hand soap, the use of dish soap, and the use of hand sanitizer. And one of the results that we came to was that the bread that made contact with dirty hands, which was our control, was the first variable to undergo changes in the texture. And that was seen on October 23rd. We noted that the change in texture, the hardening of the bread in various locations on the bread. The other control became hardened and then developed a few stale spots. And this was noticed on November 3rd, which was hardening on the crust. The antibacterial hand soap had no changes or growth and remained the freshest, and that was noted on November 12th. The dish soap and the hand soap had similar changes in texture, but did not display any growth. The hand sanitizer did have a slight effect on the bread's texture that was noticeable, and these were also noted on November 12th. And as you can see here, we have our 6 photos of the different breads. They were all of the same brand, the same type of slice of bread. They weren't touched by our hands so that we could get the best results possible, except for the bread that was touched by dirty hands.
These pictures resemble the results of after what happened to our experiment. The bread that showed the most effect to it was the bread that was touched with the dirty hands. In conclusion, we determined that hand washing is the best form of disinfecting hands. Even though that the hand sanitizer did not significantly fail, it is proven that it was not as effective as the hand soap. For future experiments, we will consider the type of bread we use based on the preservatives that might have affected the outcome of the experiment. With this experiment, we can confirm our alternative hypothesis in that the hand soaps treated bread was in fact fresh and had no mold growth at the end of the experiment, and was in fact fresher than the hand sanitizer treated bread. While the hand sanitizer treated bread showed no mold growth, it did show stale spots, therefore proving that the hand soap is the best method for sanitizing hands and preventing bacteria. However, the null hypothesis cannot be fully rejected in the fact that none of the breads that were treated saw any mold growth, and this is possibly due to, as said before, the possible preservatives in the bread or ingredients in the hand soap. And the background article that we used for our experiment was Show Me the Science, Why Wash Your Hands by the CDC.